So we copy that link that has been given for target scan human. So target scan is another database that is free and is available online.
So that's section now for the genes targeted.
You want to use this target scan. There are other databases that are also used for target gene, microRNA target gene prediction. We have like database. We have the database. We have the Miranda. But the Miranda is something that you have to input. It is a software that you have to put and then adjust by yourself. And you will need a source code, okay, from the supplier. So it will not be very good for some of us here in Africa who don't want to purchase. We want free things. Yes, we always want the free things. Why is that thing slow? Okay, my laptop had disconnected. Are we there? Yes, sir. I mean, it's only my laptop that got disconnected from the Wi-Fi. So I was saying that there are other databases. We have something like database that gives you or predicts for microRNAs or targets for microRNAs that have already been experimentally proven. We have something like Miranda. We have the Dynamicro T CDs, MVT CDs. Okay, so there are many of them. So this is the site. This is the database. Search for predicted microRNAs in mammals. Enter the human gene symbol for that microRNA. So if you look at this window up here, okay, these are the different organisms where you can do the prediction of this microRNA, and they keep adding, okay? Some other tools might just have for humans. Some other tools will just have from humans, mouse, and rat. I think this target scan is the most updated with respect to the number of organisms or microRNA in a number of organisms. So that is why we took this one. So if we want for humans, since we are dealing with the human microRNA, we select human. Okay, and they say enter a human gene symbol. Okay. So we don't want to enter any human gene symbol there. Or an assembly. So in this case, we are using microRNA. Okay. But in another case, you might have but a gene. If you have but the gene, you come and you input here. Are we together? Or if you have a group of genes, you can input the group of genes here. But us, we are starting from microRNAs. Because we are starting from microRNA, we will not input anything here. Okay? Are we seeing where I'm clicking? On that number 2, we don't input anything. But if had it been we started with genes, we'll input the genes here. Okay? Now, under they say other me-based annotations. Enter a microRNA name. E.g., they have given you this. Okay? But that is not what. Let's look for our own microRNA. That is this me 29A3P. We copy it. Now you want to type it? Okay, I think I searched for this microRNA on this laptop, so it just came up. So what do? We submit.
Submitted.
The name of the microRNA. Go to that place below where you see the name, input the name of the microRNA. That is where we have to allow all the other things on the database. Don't touch anything. Allow them at default settings. Go to where you have the input the name of the microRNA. Have you seen? Then you input it. Okay? Are we there? Then you submit. Click on submit below. Have you submitted? Have you come to this page? Okay, so we see all of the genes that are targeted by this microRNA. Okay? The total is 1,265 transcripts with conserved site, containing a total of 1,463 conserved sites and 324 poorly conserved sites. Okay? So please note that this predicted target includes some false positives. Okay, since it's a prediction tool, they will just input some parameters. Might be if the seed site, there should be a complete complementarity. If it is conserved, might be they will select that the seed site. If it is there's complete complementarity, you should select it as a target. If this selected site is conserved over different organisms, you should select it. If the free energy, so those are parameters that are selected for microRNAs. Okay, so this target will input all of those parameters into the database, and each database has its own parameters that it has selected. If you go on the YouTube the YouTube video that I had uploaded, you will see all of these explanations. Not so? Are we together? The parameters that are selected for databases. So it is obvious that it should give some false positives. That is what I'm trying to explain. So genes with only poorly conserved sites are not shown. So it's a very long table.
So these are
very long.
All the genes targeted by this.
Yes, all these genes are targeted by this microRNA. Okay, so you go right below is 1,200 and something. Okay, so we want to download. You look on the right, the extreme right of this table, you will see download table. Okay, you click on it. The extreme right, at the top of the table, you see download table. Okay, so they give you two files, right? We'll take the Excel file. We'll select the second file, which is the Excel file. Okay, you click. The second file is an Excel file. So you click on that second file. You see it downloading below in the Excel, in an Excel file. So all that 1,200 and something genes will be, so you can open the file. So these are all the genes that we have downloaded. So here we have the gene names on the third column C. We have the names of the gene, and we have the target genes, their abbreviations. Okay. So this is where we are. I always like to work with these gene abbreviations. Okay, so this is what we are going to copy. So these are all the genes. We have already done our targeted genes and we have obtained our genes. Okay, so this 1,200 and something are the genes. If you want to work with the genes, you can now look at the genes here. You pick each and you test it on the bench. If you want. Yeah, you can select anyone and test, not so, to see whether it is true. Okay, it can be a research topic. This is a whole database that we are playing with it here like this. Okay, you can take and then you select some number of genes and you test the microRNAs on the bench. Let's say you can select 10 and you test it experimentally and you confirm that, or you give your hypothesis that based on bioinformatics, it has predicted these genes to be targets for this microRNA. Okay, now you are trying to test whether this microRNA is actually the target for this gene. So it's a whole, you see a whole database or data that has been pulled out from bioinformatics that can save us as so many research topics. Okay, voila. So we'll copy this. Whose stuff is still loading? We come here and we copy. You see that with these genes, from here you can just go and test, or you can also get information on this number of genes that has been given. And how do we get this information? We can now proceed to pathway prediction. All these genes, in which pathways are they involved? Or which pathway is highly enriched by all these genes? Now it can start to give you an information that can be interpreted. Are we clear? Because with all these genes, you are looking at it, you cannot give somebody an explanation what all these genes mean. Okay? So, if we want to draw out important information from these genes, we will copy it and move into pathway prediction. Are we together? Good. So they say submit these genes to the database for annotation, visualization, and integrated discovery. The short form is DAVID. This is another database. It is an integrated database. Okay? So Bioinformatics Resource 6.8. That is the link that has been given. Use the identifier official gene symbol and select Homo sapien as the species and gene list as list type. Then submit the query. That is question 6. So we have to move now into this DAVID database. We are with question 6. Copy that link of that DAVID database that has been given and paste it into your browser. Before you copy the genes, I've said that you should copy that link for DAVID database and put it into your browser, paste it into your browser to go to the site. When you should have gone to the site, you can now come and you copy these 1,200 and something genes that we had on Excel. Has it opened? It's slow. The internet is slow. So these are heavy sites, so they consume a lot of data. That's why it's always taking time like that. Voilà. So we see some information that has been given. If you have to read or you want to read, you can read it. The database for annotation, visualization, and integrated discovery, version 6.8, comprises a full knowledge base. I told you what knowledge base is. Knowledge base update to the 6th version of our original web accessible programs. You have signed out? Okay. David now provides a comprehensive set of functions